Hello everyone, we are group 3 of the final presentation and we are going to present about sensors, bioluminescent tools, and optogenetic tools. To begin with, we'll start with sensors for astrocytes. Astrocytes are one of the major non-neuronal cell types in mammalian central nervous system, and they were thought to be electrically silent. Thus, traditionally, astrocytes were considered as brain glue or just support cells. But the development of new optical imaging tools, such as organic ion-sensitive dye and genetically encoded biosensors, has led to a dramatic change of this thought, and people began to consider astrocytes as key components and regulators of neuronal circuits. But still, there were recent discoveries of astrocyte subtypes and their functions strongly influenced by behavioral state. Therefore, tools to genetically manipulate astrocyte genes and combination of these tools with functional studies will give us new insights into diverse astrocyte functions, mechanism of controlling neuronal circuits, and subsequent animal behavior, which can eventually lead to further insights about central nervous system diseases. Now, the next topic is probes for oxygen measurements by phosphorescence quenching. The porphyrin molecules shown at this slide contain central metal atom and they emit phosphorescence light when they are excited. People used platinum or palladium porphyrins to measure the ability of oxygen, which is proportional to its level to quench emission coming from this probe. The development of utilizing two photons with antennae in these phosphorescent probes in microscope along with synthetic manipulations has led to great advance in neuroimaging of oxygen distribution. Still, several novel developments to combine this imaging with sensing environmental analytes to increase orders of nonlinearity, for example, applying 3-photon absorption, and to develop specific tissue targeting sensors can be done. Next, we are going to discuss about bioluminescent tools for imaging, modulating, and integrating. As usual, photons were generated by a physical light source, but an enzyme called luciferase can produce light by oxidizing luciferin. This bioluminescence can be utilized into various techniques, for example, finding causal interrogation of synapse, or using light-sensitive photoreceptors to perform specific function or to enhance neuronal repair, and even integrating the biological processes. Further molecular engineering of this bioluminescence can allow more sophisticated version of imaging, controlling, and integrating neuronal activities, which can be closely related to the next topic, optogenetic actuators.
Optogenetics is a technique to control the activity of neurons or other cells with light. This began with the discovery of light-sensitive protein, channel rhodopsin. First of all, optogenetics requires light-sensitive proteins. Cation-conducting and anion-conducting channel rhodopsins are the most widely used opsins, and the influx of ion leads to neuronal activation by depolarizing or inhibition by hyperpolarizing. Ion-pumping rhodopsins have advantage of high ion specificity and active transport of ions against the electrochemical gradient. Unfortunately, pumps are limited by a 1-to-1 proton-to-ion stoichiometry. G-protein coupled rhodopsin is not directly involved in ion transport, but can mediate the various signals by catalyzing GDP-GTP exchange through the engagement of heterotrimeric G-proteins. Enzyme rhodopsins can modulate the secondary messengers that mediating cellular process and signaling pathways. Genes encoding light-sensitive proteins can be delivered by transfection via transduction or the creation of transgenic animal lines. Genetic engineering techniques allows the expression of optogenetic tools only to the intended cells, and this makes a powerful advantage in delicate modulation. Accurate control of cellular activity depends on the well-defined temporal and spatial control of the light. Wide-field illumination can be controlled using an LED or one-photon laser scanning microscopy, and all of light-sensitive cells in the light path will be stimulated together. On the other hand, two-photon scanning microscopy can be used for targeted activation of single light-sensitive neurons. Electrical stimulation and drugs, which have been used to manipulate neurons, have limitations that affect not only targeted cells, but also other cells around it. With the advent of optogenetics, only desired neurons can control quickly, accurately, and safely. Nowadays, research of applying optogenetics to treatment of various mental diseases such as Parkinson's disease are being actively studied. In addition, it is expected to be used for the visual function recovery of the blind. You can find more detailed information in these references. Thank you for listening.